I'm very excited and glad to introduce our second webinar speaker, Dr. Krishna Agarwal. She is a professor in the Department of Physics and Technology at UIT, the Arctic University of Norway. We are really, I thank to agree to be a part of this webinar series. So she has received her PhD from NUS, Singapore in the year 2011 and BTech from IIT, Dhanbad, India. She has received a distinguished, nice awards that I can see from Distinguished Alumni Award from IIT. And she was awarded the Mary Sklova Curie Actions Individual Fellowship for the year 2019, 17 to 19. She was awarded the Young Scientist Award by Union Radio Scientific Research and Technology Center. She was a research fellow at NUS from 2010 to 14. She was a scientist at DRDO in India from 2003 to 6, so 2006, and she leads EU and ASEAN projects as a principal investigator and co-investigator. And it's great and pleasure to see that she has raised a fund of approximately 14 million euros. And her current research interests are computational nanoscopy, super resolution imaging, and inverse problems. So today we are glad to have you here, Dr. Krishna, and we are happy to hear your signal classification algorithm. And over to you, Krishna. Just a second, sorry. Just couple of one instruction to the audience. In case if you have questions to the speaker, I request all the participants to post your questions in the chat window, which will be taken by the moderator at the end of the talk. Please, over to you, Krishna.
Thank you, Lakshmi. Thank you, Nihit. Also, thanks Indian Bioimaging for thinking about me and giving me this opportunity to present musical multiple signal classification algorithm. As Lakshmi said, I am a professor in UIT, the Arctic University of Norway. We are latitude-wise really the northernmost university of Norway. I am sitting here in the Arctic Circle in my office to talk to you all today. Um, and the topic that I'm presenting is actually a versatile super resolution technique called musical, which I invented in 2016. And now it can be used for a variety of applications, as I will demonstrate. But before that, a little bit of introduction of my activities and where I am and so on. So I lead this group called 3D Nanoscopy Group, and we are just like I was saying above the Arctic Circle. And what do we do there? We do computational microscopy and nanoscopy for living biological systems. So as compared to a lot of work in microscopy development, not applications, but development, our core focus is living biological systems. If we can develop new techniques, new solutions for living biological systems, that makes us very happy. And when we say that we work with living biological systems, we talk about fluorescence imaging. So the majority of this talk today is dedicated to fluorescence imaging. But a lot of my recent work is actually about developing label-free nanoscopy, which is label-free super resolution microscopy. And this is very exciting because it allows us to do quantitative imaging. However, these techniques are really very, very new and they are not at present available as commercial solutions. So that is the reason I didn't want to talk about label-free imaging today. To Indian bioimaging community, which is a community which thrives on existing commercial infrastructure in imaging labs. My collaboration spread all across the globe, and all my research is funded through primarily European Union and the Research Council of Norway. But I have also received generous funding from the Directorate of Higher Education in Norway and the Center of Digital Life Norway. Um, we are a thriving group. We have a lot of new research as well as new innovations going on in our group. And that calls for, of course, a very multidisciplinary team of 20 members. So they are the force behind me, and they are behind a lot of the content which is being presented today. When we talk about super resolution, we usually it's very natural for us to think of super resolution as shown here, that there is something which human eye can see, almost like the size of a hair, maybe something slightly smaller than hair, and beyond that we need optical microscopes. And then there is a limit to which we can use optical microscopes. Even the best ones cannot go beyond a certain limit, and then we need something more. Of course, we can use electron microscopes, atomic force microscopes, a variety of other microscopes, but they are destructive in nature. So, this philosophy of catering to living systems is not supported by them in general. And that is where people thought that we should still be able to use optical microscopes but push their limit. So, this is the usual way of thinking about it, and I think it is not wrong, except that when someone in from the physics background thinks of super resolution, they think of it slightly differently. And I want to provide that perspective to you because it can help you use super resolution techniques in a more broad scope than visualizing proteins or visualizing something which is molecular. And this perspective that I am presenting is now coming up in this slide. But let us see what super resolution microscopy can achieve. So here I have a wide field image of liver endothelial cells. These cells are between the bloodstream and liver tissue. So these are the blood barrier cells. And the function of liver is to filter. So basically, these cells are performing some kind of filtration. They have something called nanopores, which are between 100 to 250 nanometers big. And these cells are basically like sieves, like colanders with these nanopores. And they allow or restrict molecules and viruses of certain size to pass through. So in the wide field microscopy, we do not see the sieve-like structure, the nanopore structure, but when we apply super resolution, we do see the sieve-like structure. That is the strength of super resolution. And of course, this was so phenomenally empowering and so much beyond the scope of our imagination that it actually received Nobel Prize. So why we cannot see beyond a certain limit in a microscope is itself a very good question, a very good starting point. Let us say we have fluorescent molecules which are arranged in a particular configuration, and they can be placed like pretty close to each other, and each molecule, of course, is of a very small size. But the microscope itself produces a blurred spot for each molecule. And this blurred spot, if you are really lucky, is approximately 250 nanometer in diameter. If you are not so lucky, if you don't have the best microscope, if there are aberrations, if there are other problems, you will see each molecule as a blob of a much bigger size. So what does this mean is that your image essentially looks like a very, very blurred version of the reality. And if you look at this blurred version, I don't think any of you would predict that there is actually a star which is underlying it. It could as well have been a more flower shaped structure or something different, right? And the view that physicists take is that super resolution optical microscopy is to break the curse of the instrument. If you look at an instrument, including a microscope, it shows a projection of reality. And in the microscope, this projection of reality is significantly blurred. But when someone is asking a biological question and using microscope as a tool, they want to be able to estimate reality better than what the tool supports so that they can make useful conclusions out of that. So the trick in this super resolution microscopy is to actually make the measurements more informative and then to be able to decode this information in a way that we can exceed the limit of the instrument. So let us say you have a 10x microscope with zero point 0.4 numerical aperture objective lens. maybe your resolution limit is 1.2. I'm just throwing this up. This is like I don't hold me to this number 1.2 micrometer that I'm saying, right? But if using super resolution technique, you can use the same instrument to reach, instead of 1.2 micrometer, maybe something like 600 nanometer or less than 1 micrometer, then you are already achieving super resolution. So super resolution is not about going less than 100 nanometers. Super resolution is about surpassing the resolution supported by the instrument. And with this slightly gentle introduction to how physicists think about super resolution, let me bring up this slide which provides a very large picture of what is happening in super resolution. What are the solutions that you have? This is by no means exhaustive, but it is a representative of all the major things that are happening in super resolution community. There is structured illumination microscopy. This is a very robust form of super resolution microscopy. Here you use these patterned illumination or illumination patterns, which allow us to see nuances of sample 2 times smaller than the resolution limit of the microscope. So it's not like it's resolving to a great extent. There is only a factor of 2, but it is very reliably achievable. However, this reliable achievement comes at the cost of very expensive instrument, extremely sensitive calibration. You are off in your calibration just a little bit, and your images will have a lot of artifacts. So, alignment sensitivity, calibration requirement, extremely expensive instrument, but very reliable two-factor enhancement of resolution. Then we have stimulated emission depletion. And this is actually also a very cool technique because it uses confocal microscopes base, but instead of using a single intensity or single light for taking one confocal image, it uses two lights. And these two lights are selected in such a way that one light can actually deplete molecules of a certain kind. So we choose fluorophores. These are not regular fluorophores, these are stud fluorophores. And then we introduce these stud fluorophores into the sample, and we know that all of these fluorophores will go quiet if I use a particular light. So then in the confocal system, we have the original illumination, but we also have this extra light in donut shape. So at the overlap of these two beams, there is a very small spot which does not have the depleting laser light. And this is how you achieve actually an illumination spot of 20 to 50 nanometers, depending on the optics, right? So this means that instead of confocal's resolution of approximately 200, 250 nanometer, you can actually achieve resolution of 50 nanometer, even 20 nanometer. And then there is, so this is again, STED again uses an expensive microscope, which also needs to be upgraded for this extra light and its alignment and other things, but it does not involve any computation. SIM does include computation, and that is why calibration is important. There is another technique called single molecule localization microscopy, and that is the beauty of that is you do not need to change your microscopes. You do not need to have more advanced equipment. You can actually use a regular wide field microscope to achieve single molecule localization. But then it uses fluorophores, which exhibit a very certain property. We are coming to that very quickly. And this property allows us to separate molecules from each other, which then allows us to make a super resolution image. But then there is this emerging community of fluctuation based microscopy, and this is actually very close to single molecule localization microscopy, but it beats many challenges that single molecule localization microscopy uses. And musical, the method that I'm presenting actually falls in fluctuation based microscopy. So with this, I would really like to quickly go into single molecule localization microscopy because fluctuation based microscopy builds directly on top of it. In single molecule localization microscopy, we use these very specific fluorophores, which are called single molecule fluorophores. There are palm fluorophores, there are paint fluorophores. All of these fluorophores are used for single molecule. And these fluorophores show something like a blinking phenomenon. Let me show to you what this blinking phenomenon looks like. So if you have a very high quality single molecule data, you would see a video which looks like just stars blinking. You would not see the structure of the sample. You would not see many details. You would only see these blinks. And the reason is that these fluorophores, they are emitting light only probably once in 1,000 or once in 10,000 instances of illumination. And this is happening randomly. So it's almost like we ask a question in a big classroom, and there is usually students speak less because they feel on the spot, but someone says something, and then we ask again, and someone else says something, and so on. So it's almost like that. Now that we make such a video, we can actually computationally find their centers because we have these blinking fluorophores. We see almost one spot at a time, and we can find the center of the spot using an algorithm. And then we can keep on collecting these spots one after another frame. And eventually we see the image where we have single molecule spots for the entire sample. So this is the concept of single molecule localization microscopy. No instrument modification, yes. Simple computational processing, yes. Molecular resolution, yes. Three things going for it. But it requires thousands of frames if you are lucky. If you are not so lucky, it requires close to 100,000, sometimes even millions of frames. And it has challenges with photon budget. So these blinking fluorophores, they have, first of all, a more limited photon budget than the regular fluorophores, which means that they quench or oxidize easily. And also, many of these dyes initially were not life cell friendly. There are now new set of dyes which are more life cell friendly, but they are still not natural enough for the cells to feel to not change their natural behavior. So with this as the limitation, fluctuation based microscopy asks a very simple question. Can I actually achieve super resolution without changing the instrument with only a few hundreds of frames, maybe even less? And can I use regular dyes which have been proven in fluorescence microscopy more and more? And with this as an inspiration, a lot of new methods have been developed. Before musical, there were three methods which came up: SOFI, 3D, ESI, and then musical, and there is another technique called surf. They came up almost simultaneously. And after that, there has been some development. The most recent one is MSSR. And they all have different characteristics. Um, but the ones that are really nailing these two needs are generally musical and surf. And now I will focus more on musical because that will help me give you a tool which you can use directly in your labs. But if you really look at all of these techniques, first of all, are computational because they don't change instruments, they don't change dyes, they have to do something somewhere else for super resolution, and that is computation. But this computation is not like finding centers of blobs. This computation is some kind of advanced statistical analysis. And here I have color coded these techniques according to what statistical analysis they use. Musical stands out in comparison to all of them because musical checks for only one thing, and that one thing is randomness over space and time. What is random over both space and time? It's just noise. So what musical is testing for is something noisy, is a pattern noisy? If it is noisy, we suppress it, and that is how musical gets super resolution. It's a very, very simple concept. So in musical, we again label this sample with fluorophores. We put them under a regular microscope and we take a video. The key reason or the key philosophy of musical is that noise is the only thing which is random over both space and time, which means that in a frame, if you are looking across space, if there is something really random, it's noise. But if you look at a particular pixels over time, then also there is noise which is random. On the other hand, if it were a regular fluorophores, which was labeling a sample, the fluorophore would show up in each frame. and the fluorophore would show up in each frame through this point spread function. So the point spread function is contributing to spatial non-randomness or special systematic pattern, right? But also, fluorophore is not emitting the same number of photons every time. So there is temporal fluctuation or temporal randomness contributed by fluorophore emissions. But there is spatial encoding through the point spread function of the microscope. And this is the information that musical uses to separate noise from fluorophores. We can discuss a lot of mathematics. I will go through these next 2 or 3 slides very quickly just to tell you what happens, but not to overwhelm you with algorithmic details. Musical uses eigenanalysis. Eigenanalysis is a statistical technique that is one of the first techniques that founded machine learning, the old school machine learning, not the new deep learning. And through eigenanalysis, musical finds or computes something called eigenimages, and it can sort these eigenimages into images which have some systematic pattern and images which are more or less noisy. And when we take these images of these two categories and we perform something called which is so using this computational confocal scan on these two sets, we compute two images, one representing all the information in the signal set, the other one concisely presenting all the information in the noise set. Now, this is very interesting because the this signal set produces an image which is mainly denoised and deconvolved. It is much sharper than the original one and it does not have background noise. But the other one, the one that is computed using this noise set, is very interesting because it makes zeros at positions where fluorescent molecules are present. Why does it make zero? Because that those are the only points where the signal is not random. Everywhere else, there is randomness because of noise, but at the fluorophore location, there is a systematic signal coming at every time point. So the noise image actually has zeros at the location of fluorescent molecules, and this allows us to identify the location of fluorescent molecule or the distribution of fluorescent molecules. So what we do is, in order to turn these zeros into kind of a positive image, we are usually used to seeing positive image. We take a ratio of these two, and due to this ratio, first of all, we get this super resolved image where we have high points at the location of molecules, but we also use this denoising and deconvolution properties in forming the final image. Let me give you an example. So mitochondria was labeled in a living cell, and the mitochondria was labeled for its inner membrane, which labels cristae. And we used 5 millisecond exposure time, wide field microscope 1.42 Na, and we took 200 frames. And if you look at its averaged image, which is like high quality diffraction limited image with long exposure, this is what you get. But if you apply musical, you actually do see cristae structure. So this is the power of musical. But it's good to do a sanity check about using some kind of benchmark, some kind of gold standard. So we also use the EPFL SMLM data, and we saw that musical can produce actually exactly the same quality images as single molecule when the data has single molecule properties, which is the blinking properties. And in the situations where a single molecule starts failing because there are many molecules which are emitting light together, their musical actually outperforms single molecule techniques. So the sanity check says that, yeah, musical is not imagining structures, it is actually creating structures which are reliable. Now, the versatility of musical comes from the fact that it is just checking randomness over space and time. It is just checking for presence of noise. And the good thing is that this presence of noise happens in any fluorescence imaging data irrespective of whether the fluorophores were blinking or not, whether you had patterned illumination or not, whether you had 10 other conditions or not. So this means that in principle, musical can be applied to any noisy fluorescence video set. And this is a very liberating concept. This is what makes musical versatile. We have checked musical for a huge variety of conditions, and we know that musical works for 2D sample, for 3D sample, for sample which is sparsely labeled, for sample which is densely labeled. It can work for any dye, fluorescent proteins, and so on. It can even work for label-free autofluorescence. And it can work for a variety of other photonic phenomena like two photon, like Raman, and so on. It works for any wide field system, but it also works for confocal systems. And it can work with any source of fluctuation. Like I was saying, that single molecule uses this blinking phenomenon where the molecules are not emitting light most of the time. So this is fluctuations due to fluorescence, but musical can use fluctuation due to illumination, which means it can be used for sim systems. It can also use fluctuation of motion, which means it can be used for nanoscale motion tracing. And it can be simply applied in extremely noisy fluorescent videos for denoising. There are some minimum algorithmic needs which are needed for ensuring that musical works well. And I have listed them there. So since this is being recorded, and if you're interested, you can always come back to them, and you can also um, drop an email to my team or me so that we can help you a little bit more. Now, let me show you some actual examples of musical and how it applies over cells, tissues, and even organism. So we wanted to do multi-scale imaging and see whether musical can achieve super resolution at different objective lenses. This is a sample which is fish scales of salmon. So salmon is an Atlantic fish, a cold water fish. And then when we clean salmon, the fish skin cells kind of go out, and we collected them, we put them under the microscope, and it's a very densely packed cell sample, and they have been labeled for actin. If you look at diffraction limited 10x image, this is how it looks. But if you apply musical on it, it of course performs denoising, but it also enhances the structural continuity of a lot of the structures. This is actually comparable to a 60x diffraction limited image. And if we apply musical further on 60x diffraction limited image, we get further resolution enhancement. Now, what is also interesting is that musical not only provides super resolution because of labeling and the noise, but if your equipment had certain aberrations, for example, this one had line aberrations, they also do get enhanced. So this means that on one hand, it creates or it enhances artifacts appearing from an imperfect system. On the other hand, it allows you to separate the image quality degradation which is happening due to aberration versus what is because of the sample. So this is another feature of musical that can be used in the applications for a more unbiased analysis or unbiased interpretation of biological images. This test of musical was done in University of Southampton, completely independent of us. They are collaborators. They have the same file that you see in the last slide of musical, which is the open source version of musical. And they tested musical on human neuroblastoma cells, and this was the confocal image. So this time it's a confocal system, and after musical processing they got this image. So this is what musical could achieve for confocal system on neuroblastoma. We also did it on tissues, and tissues are very notorious because they have, first of all, taking the fluorophores to the interior of tissues is very difficult. But also tissues are such a dense living sample that uh, when you are successful in labeling, fluorophores are everywhere. It's a very, very densely packed system. And There is no way you can make single molecule work for this. Your almost only hope is SIM. But when we applied musical on a diffraction limited video of this heart tissue, we could get actually an image which was better than SIM, and it could reveal all the sarcomeric structure of this heart tissue sections. Remember here that I'm applying musical on diffraction limited image. I will also show examples where I applied musical on the raw SIM data, and there the results are even more beautiful. Now, this is actually a living fly, and this living fly was clamped under the microscope, and its antenna was labeled with Alexa fluorophore, and neuronal imaging was done. So this is on a living organism on antenna, which is kind of much bigger length scale than your regular cells. Again, this was done in University of Southampton, completely independent of us, and they achieved a very beautiful resolution enhancement using musical. So musical does work over all the length scales that one may encounter in microscopy. It also works fantastic on patterned illuminations. In favor of time, I'm showing only this. So with this, I'm showing two things. One, that musical works on 3D samples. The second, musical works on direct raw data of your SIM microscope. So we have this SIM microscope from, uh, sorry, it just escapes my mind right now. But this SIM microscope, it was one of the, first high quality microscopes that we bought in 2014. And it has, of course, its proprietary software. And we imaged kidney organoids under them, kidney sections or kidney organoid sections under them. And the commercial proprietary software of SIM gave this image. And you see these hexagonal artifacts that you actually see in SIM when you are imaging over large field of view or over thick samples because of the modulation in consistency of illumination. We for this patterned illumination, we compute something called this modulation factor or modulation constant. And this modulation constant changes over the field of view, which introduces low quality illuminations outside the center region. Also, if you require SIM to work, you have to have a high signal to noise ratio. If you push down the signal to noise ratio to something like 2 or 3, SIM usually doesn't work. And it provides pretty artifacty image like the one shown here. When we applied musical on the raw SIM data, it was devoid of all these hexagonal artifacts, but also it provided a much better optical sectioning than 3D SIM, commercial 3D SIM. Um, and this is what I was saying, the modulation contrast was non-uniform. So there is a high modulation contrast here and low modulation contrast outside. This means that your SIM results will be pretty good here, but they will not be great around this region. So in this particular case, 3D SIM provides pretty artifacty image again, whereas musical can actually produce much more clinically relevant, high quality image, which can be benchmarked against a scanning electron microscope. This set of experiments were not done in our facilities. They were done in a German university clinic, the University Clinic of Erlangen, again, independent of us and independent of our system. Yeah, this is a little bit of zoom in into two regions. The diffraction limited image is, of course, hopeless. The SIM image does show the nephron structure, which is typical to the kidney tissue, which is used here. But musical in slightly different configurations preserves all the structures while reducing many artifacts that are present in the SIM reconstruction. In this case, musical was applied not on diffraction limited data, but your raw SIM data. So if you have raw SIM data readout, you can apply musical directly on it and get these images. Musical also works on label-free data, and I show clinical autofluorescence results here. So what we had was oral mucosa tissues, and these tissues were actually collected in Kolkata, in one of the hospitals in Kolkata, and I think there were 30 patients or something which contributed to the sample. And there were early cancer patients, there were advanced cancer patients, and so on, all for oral cancer. And this study was done using a very simple lab-based fluorescence microscope, which had just a halogen lamp and only a color filter in the collection path. So it's really a very, very basic microscope. And these were patient tissues. These patient tissues were not fixed. There was no fixation, etc. Just the patient tissues were taken. They were sliced into through a microtome into thin slices, and these thin slices were put directly under the microscope. So almost no processing, no sample processing, a very simple microscope. And the idea was to use musical for estimating the density and identifying new um, readouts, density-based readouts of collagen and keratin, so that they can indicate the stage of cancer. And how does it connect to super resolution? So what we did was a very simple numerical study where we simulated fibrils of different densities, and we computed what happens if we have a super resolution technique. Um, how does it compare with the actual density versus if we were using a diffraction limited wide field microscope? So the principle is established that if you are able to successfully use a super resolution technique, the density readouts from a super resolution technique will far better match the actual density in comparison to diffraction limited image. Now, with this principle established, we took diffraction limited autofluorescence images of the samples, and we also collected videos and we applied musical. And these are the clinical staining results of similar samples. I think the same patient, but different regions of the samples. Now, what clinicians were able to do is they were able to look at diffraction limited image and look at the intensity to kind of make this boundary between epithelium and subepithelium. They were able to do it for all the tissues very naturally. So autofluorescence density in diffraction limited images were sufficient for them to separate visually the epithelium and subepithelium layers. But when we applied musical on them, it kind of enhanced the features which represent nanoscale features of the cancer development. And what was done was that after separating these, there were small crops taken from the musical image, and for these small crops, the density was computed. And then a statistical measure of all these densities from crops in subepithelium were taken, and statistical measures in the epithelium were taken, and they were compared. So through this, so this was, I think, the ratio of density in subepithelium divided by to the densities in epithelium. And with Musitaf, using such a simple readout, we were able to identify or classify the different stages of cancer. And this was much better than using the regular histostaining samples.
So, Dr.
Uh,
Krishna, sorry to
yes,
interrupt. So, you have another 5 minutes.
yes, I think I will not even need 5 minutes more.
Okay, thank you.
There is another thing which can be interesting, and this is like if you have vesicles or small proteins which are in motion, then if you take a video and do the conventional tracking, then you get these isolated kind of spots in the track, which are then connected over time. But if you apply musical on them, you get this nanoscale continuous motion trace. Because musical is available as an open source on Image J, which is actually continuous, even if we have taken discrete videos in the form of frames. This is the example. So we have a living cell here. It's a cardiomyoblast. And we have mitochondria labeled in green. We have vesicles labeled in red. And these vesicles are lysosomes. And here we see that a lysosome moves towards a mitochondrion. But when we apply musical on this video, we see this motion trace, which is much finer. Now, we split this 400 frames into two sub videos. Two sub videos, 200 frames each. And we saw that actually there is, of course, vesicle moving towards it, but then vesicle spends a lot of time around this mitochondrion, almost stationary in that location. So this is how we can use musical also to analyze motion traces. This is the recap slide. In essence, if you meet these criteria and if you make your measurements in a way that these criteria can be met, musical can actually be a very versatile technique for a lot of varieties of samples, microscopes, and also sources of fluctuations. With this, I end my talk. And musical is available as an open source on ImageJ. You can use this QR code to download musical and install it in your ImageJ for your first tests. You can also contact me on this email address, or you can look at our team and our work on this website. Thank you very much.
Thanks, Dr. Krishna. This was really exciting. So, Nihit, can we take the questions from the participants? I think there are
Yeah.
few.
Yes. So thank you, Dr. Krishna, for this very clear and excellent talk.
Thank
We
you.
have so far two questions from the chat, but I would also like to invite other participants to post their questions in the chat if they want to.
Yes,
So the
and
first question,
yeah, go ahead. Yeah, please
sorry.
go ahead.
So yeah,
Yeah.
Krishna, if you can read the questions and if you would like to answer,
Yes.
then please go ahead. Yeah.
So in all the experiments, so the question is what kind of blinking fluorophores were used in these experiments? There were no blinking fluorophores used. In most of the cases, we used regular dyes, so MitoTracker green, MitoTracker deep red, LysoTracker blue, I think, and then there were some in which we have GFP. I have not shown images for YFP, but we also have used yellow fluorescence protein. And in a lot of them, when LXR dyes were used, which can be used for blinking, we didn't introduce the medium which kind of forces blinking. So this means even if we were using fluorophores, which can be used for single molecule, we didn't force them into blinking. They were naturally fluorescent. The second question is, does the localization density affect noise detection and are they related? Yes, the localization density. So if there are too many molecules packed together, then the spatial noise will stand out more easily because a lot of fluorophores packed together will give you a higher average illumination intensity, right? But what it will also influence is that the fluorophores would not stand out from each other. I didn't go into mathematics a lot, and I usually provide an analogy of a rice bowl. So in single molecule localization microscopy, what happens is if you were given a rice bowl and you were asked to take out single rice grain at a time, you could actually count the rice number of rice grains very precisely, but you would have to kind of go through a very long process, right? Instead, if we were using a small spoon and we were kind of taking out one scoop at a time, and we know what is the average number of grains this spoon can take, you will find something very interesting. So the average number that can be scooped out with the spoon is roughly the same. We can, if we just count how many spoons, we can say, okay, these were the, approximately so many grains were there. But because the number of grains in each spoonful is different, maybe the first spoonful there were 100, maybe the second spoonful there were 95, and so on. So in this sense, every spoonful is having slightly different signature of your molecules, right? And this is what is happening also when we take frames. Every frame has a slightly different signature of the number of photon emissions from molecules, right? So basically, if we have a very dense sample, the average will be very high and the fluctuation will be very small. And this will affect how much individual signature of each fluorescent molecule can be traced back. So denoising will happen, it will happen with a great quality, but super resolution may be restricted. So this is how the fluorescence density influences noise detection and also super resolution. How to do musical process? Please use this QR code or look for musical on ImageJ. You can install the plugin into your ImageJ, and all you need to provide is numerical aperture, and the pixel size, magnification, and I think the wavelength. That is it. So you don't need to provide a lot of information like what was camera noise, what was this, what was that. Remember that if you are using confocal and if your nominal numerical aperture was, say, NA, then when you give the NA in this particular plugin, give root 2 times NA or slightly higher NA than the actual one. Because in the confocal systems, we have the illumination NA and the illumination objective lens and the collection objective lens. So there are two numerical apertures which are playing together. So this means that the effective NA of the system is higher than the NA of your objective lens. Is this musical only software based plugin or do we need some kind of hardware? No, you don't need any kind of hardware. You typically need videos, or if you have a sim microscope, you need the raw data from the sim microscope. Um, Anything else? Nihit, you are muted. You still are muted, just in case you are talking to us. Oh, Santosh, yeah.
Sorry. Yeah, there was some issue, but it's okay. It's okay now. So I would like to request other participants to please put their questions in the chat, or if they want to just unmute themselves and ask questions, they can also do so. In the meantime,
Or
I had
email me.
So,
or email Dr. Krishna. That would be nice.
so Nihit, I have a question. Can I?
Please go ahead.
So Krishna, have you tried musical in EM images?
No, musical does not work in EM images because in EM images,
Okay.
uh, we do not have a concept of point spread function. So this
Right.
point spread function is
Exactly.
giving you the spatial consistency or spatial
Mm.
encoding. So this is missing in electron microscopy images. Yeah.
So then, non-fluorescent images, say bright field or phase contrast, how does it work?
Yes.
Does it work there?
Yes, I avoided showing those results because we are actually in the process of commercializing musical on
Okay.
bright field and DIC and phase contrast and so on. Yeah,
Okay. Okay.
but it works with some
Okay. That's
tricks, with some neat
yeah.
tricks. It
Good to know.
works. Yeah.
Hope to learn that soon. Thank you.
Thank you. Yeah.
Thank you. I also had a small question. It's about when you mentioned that musical can also outperform storm imaging to some extent in some cases. Can you please explain how does it do?
Yes. So storm or any kind of single molecule technique works if this blinking statistics or blinking phenomenon is observed, right? So essentially, let me try to get back the slide. I hope this comes up. Just give me a second. Mhm. Yeah, this is the slide. Um, I hope you see this video.
Yes.
Yes. So the expectation or the assumption of storm is each of these blobs or these starry things that we are seeing corresponds to only one molecule. Now, most of the cases, if you have a good blinking sample and the sparsity of labeling is low, then you can achieve this condition. So what happens in these cases is that the fluorophores have duty cycle of, let's say, 1/1000, which means for 1000 photons it receives, it gives out photon only once. So this means that probably 999 fluorophores around it are silent when it is blinking, when one molecule is blinking. And this assumption is very important for the algorithm of storm to work, because then we say that really at the center of each of these blobs, there is a molecule, right? But as we increase the density of fluorophores, or we do not have a high duty cycle of blinking, then this condition is violated. And when this condition is violated, each of the blobs may correspond to a family of molecules. And in this case, your localization becomes more and more inaccurate and imprecise. So this is exactly what happens in the benchmark example that I have included, the EPFL's example here. So this particular benchmark example that you see, it has tubulin molecules, and these tubulin molecules are pretty sparse. Also, the fluorophore that was used here was a very high duty cycle or very low duty cycle fluorophore, which means that you really see each blob corresponding to one molecule. This is, by the way, reconstructed from the video that I was showing just now. But this one was tubulin with high density. And when you have tubulin with high density, then basically the assumption that each blob corresponds to one molecule is broken. And that is when storm starts giving blurry, imprecise results. And if you do deconvolution before applying storm, even then it gives not so sharp images. Does that answer your question, Nihit?
Um, yes. So basically, it's the difference between how you reconstruct the images, and that's why musical wins
Mm.
for a dense sample.
Yeah, exactly.
Okay. Thank you. There is another question in the chat, so perhaps you can go ahead.
Yes. Actually, this is a question which, um, okay, I really need to. Here it was. Yes. So the question is, how does it compare with 2D and 3D samples? Is that correct? The question, as I read it, is what are the limitations of musical technique with thin and thick samples? So, with thin samples, musical doesn't have much limitations because in thin samples, the point spread function can be kind of estimated more accurately and so on. And with thick samples, we can discuss about how thick we are talking, right? So we can have something which is one cell thick. So, which means that we need some kind of confocal or bright field to see the entire depth. Um, we cannot use turf if we want to see the entire depth. But then we can also go to organoids, right? And organoids, etc., they can be anywhere between 30 micrometer to even 500 micrometer, right? And what we have seen is because musical uses this point spread function, it works typically well for samples as thick as 10 to 15 micrometer, where the point spread function estimate is sort of reliable. And after that, the point spread function estimate gets, or point spread function actually starts aberrating. Why does it start aberrating? Because when you are out of focus, or not out of focus, I don't think I'm using the right word here. So you have a thick sample, and then there is an illumination. If you are not using confocal illumination, then basically the entire sample is illuminated. It's an epifluorescence illumination. And if it is epifluorescence illumination, there is a lot of light scattering from the part of sample which is not in focus. This light scattering creates aberrations in the point spread function. So these aberrations are relatively small when your sample is as thin as 10 to 15 micrometer. But if you have much thicker samples and you have non-confocal illumination, they become overwhelming. And in this case, musical may start giving some artifacts. So this is one tip. The other thing is if you want to really apply it for even 10 to 15 micrometer samples and you want to do 3D, then you really want to apply musical plane wise instead of applying it across the entire volume together. So that is the second, it's a tip or a trick rather than something which is limiting. And then basically for extremely thick samples, we can augment musical by actually estimating the scattering as a part of the process. Again, this is something that is still not published and not released in open, but we are working on it. So at present, thick samples, maybe 10 to 15, very reliable. If they are much thicker than that, use confocal. So when we use musical for this fly, we used confocal because, or confocal was used to avoid the problem of, the problem of illuminating everything and having scattering from everywhere. So for thick samples, use confocal.
Thank you, Dr. Krishna, for answering the questions. In the interest of time, we would like to now close this webinar session, and I would like to ask Lakshmi to please conclude. Thank you.
Thanks, Nihit. So I would like to thank once again Dr. Krishna for the wonderful lecture and nice, insightful musical application. Actually, it's going to be tremendously useful for a lot of people. Um, so thanks everyone for showing interest and attending the webinar. And so I would like to thank IBI for uh, giving this platform and hoping to keep us watchful in the social media handle for more next webinar and announcements on the same. Thanks everyone.
Thank you.